MedCram.com.
Welcome to another MedCram COVID-19 update. It's been a little while because I've been in the intensive care unit treating COVID patients. Sorry to say that we are starting to expand a little bit in Southern California, and we are seeing more patients now in the intensive care unit with COVID-19. If we look at daily new cases in the United States, they are certainly on the uptick here. And whereas we had not seen an increase in the new daily death rate in the United States, it is now starting to increase. So where I work, which is in San Bernardino County, which is in Southern California, since the beginning of the pandemic, what's happened to hospitalizations, you can see here, the green graph is the confirmed COVID-19 positive patient representation, and the pink here on top are those that are suspected. You can see overall that hospitalizations here in Southern California are on the rise, and that is not unique. In terms of the United States, we're starting to see daily new cases in South Dakota start to reach all-time highs, and the death rate is also going along with that as well. We're also seeing that in North Dakota as well in terms of daily new cases, and also the daily death rate as well. But perhaps the epicenter here in the United States would be the state of Texas, where we can see that daily new cases in Texas has reached an all-time high, but daily deaths have not yet increased. That could be because of a time lag. Of course, this is not just the United States, but it's going up around the world. We see daily new cases increasing here and are at an all-time high. And daily deaths around the world are also at an all-time high. As an example, daily cases in the United Kingdom are quite elevated. And whereas before we did not see deaths increasing, we're starting to see daily death rates increase in the United Kingdom. In Italy, we are again seeing an increase in the daily new cases. And this is reminiscent of what we saw in March in northern Italy, where we are seeing an increase in daily deaths as well at this point. And this is all to say that, of course, there's a lot of bad news around the world regarding this virus. And of course, recently there's been some good news, and we're going to talk about vaccines. And a great website to follow this is RAPS, or Regulatory Affairs Professional Society, which has a great COVID-19 vaccine tracker. And for the next couple of videos, we're going to highlight different vaccines that are being developed around the world for COVID-19. In the next few months or even weeks, there's going to be vaccines that are going to be released, brought to market, and consumers are going to have to make a choice as to which vaccine they want to get. It's going to be really important to understand and know about those vaccines and their track records. So we're going to dig deep and look at these vaccines and compare them looking at various characteristics, safety data, and efficacy. And the first one that we're going to look at is the vaccine that is being put out by Pfizer and BioNTech. And you can see here on the COVID-19 vaccine tracker, there are a number of different vaccines that you can click on them, and there are links so that you can look at the different publications and be educated on these vaccines. So the first thing that we're going to look at is the Pfizer-BioNTech one that is here, and let's get started. So the first thing that you've got to understand is the biochemistry of what's happening with an infection. Now, as we've talked about before, you have a cell, and on that cell is a ACE2 receptor. And then what you have is the virus, and on the virus, as you've heard by now, are spike proteins. And so what happens is this spike protein interacts with this ACE2 receptor, and the virus gets internalized in the cell, and it basically takes over the cell or hijacks the machinery of the cell. And so really, this interaction between the spike protein and this ACE2 receptor needs to be blocked if you want to prevent infection. And one of the ways of doing that, traditionally, is by injecting fragments of the protein into the body and have antibodies be made against these spike proteins so that the virus is not allowed to bind this ACE2 receptor and you neutralize this infection. And that's generally how vaccines have worked in the past. And the way that this virus, SARS-CoV-2, how it does this is that there is this mRNA genome which is inside of the virus. And so when the virus binds with the cell internalizes the envelope, but it also internalizes the messenger RNA inside the cell. Now, if we were to blow this up and show you what happens inside, it would look something like this. Here's the cell with the nucleus. In the nucleus, you have DNA, and that DNA is converted into RNA. And these are made up of nucleotides. Well, that RNA is converted into messenger RNA, and it's exported out of the nucleus into the cytoplasm, where you then have something called messenger RNA, which is still in the same language as the DNA is. But now you've got mRNA. And mRNA is a very specific structure. It has something we call a 5' cap. It has information, and then it has a non-transcribed region at the end with a poly-A tail. And this is what allows you to make proteins, because this messenger RNA is translated by something called ribosomes into protein. And it's this protein that can turn into just about any type of protein you want, depending on the instructions here in the mRNA. Now, as we talked about before, when SARS-CoV-2 infects the cells, it causes the machinery in the cell to make proteins for more viruses. And so it's making more spike proteins, it's making more proteins that go on its surface, it's making a whole bunch of other things that go into making more SARS-CoV-2 virus. And of course, these proteins here can be incorporated into the cell membrane, or they can be exported to outside the cell. So this sort of brings us to why Pfizer and BioNTech are teaming up. It's because the German company BioNTech has perfected, or at least significantly advanced, the technology of infecting with messenger RNA. And so how this new vaccine is going to work is they have a way of making messenger RNA, but delivering it to the cells in a lipid membrane. Now, if you imagine a micelle, or a lipid bilayer, in which the mRNA is enclosed into, so it's well protected, these very small particles can then fuse with these cells, and then what happens is the messenger RNA that they have engineered goes into the cell's nucleus here, and that engineered messenger RNA will then create a protein that they have genetically engineered. And in this case, that protein is a version, if you will, of the spike protein. But it's a version that is slightly different in that it holds the position of what it looks like when it is docked outside the SARS-CoV-2 virus, not after it binds and is internalized into the cell, so that the final product of this messenger RNA, after it goes into your cells, and after your cells take their messenger RNA and make the protein that they want your cells to make, it's going to make a protein inside of your body that is going to be immunogenic and cause you to form antibodies against this spike protein. And here's what it looks like. This is a publication that was published on bioRxiv, and we'll give you the link to it in the description below. But you can see here that this is the messenger RNA that they've constructed. Here's the 5' cap analog. Here are the coding sites for the receptor binding domain. That's where the spike protein binds to the ACE2 receptor. And here is S1, S2. These are portions of the spike protein. And then you can see here is the non or untranslated region. And then finally the poly-A tail. These additions to the messenger RNA allow it to escape destruction in the cytoplasm of the native cells. So in other words, it preserves it so that it gets translated into the protein. And here's a picture of the protein here down below that your body makes from the instructions from this vaccine. It's a very immunogenic structure that causes antibodies to be made. Now, last month, in the New England Journal of Medicine, they published an article called Safety and Immunogenicity of Two RNA-Based COVID-19 Vaccine Candidates. And these were the two candidates that were put forth by Pfizer and BioNTech. And they wanted to see basically how these two candidates did to see which ones would go on to the phase 2 and phase 3 trials. So let's take a look at these, because one of the big questions that we're going to have when we take a vaccine is, how safe is this vaccine? And while we're not going to get a full picture early on because a lot of the data takes months and months to come out, we can certainly see up front what type of a profile these vaccines have. And you should know what the names of these two candidates are. The first one is BNT162b1, and the second one is BNT162b2. So we'll call this number 1 and number 2. And basically what they found was, while they were not looking for efficacy, they were looking for safety, they found that the second vaccine, number 2, was associated with a lower incidence and severity of systemic reactions. So the first thing we're going to do is look at some of the side effects, and we're going to look at both vaccines, both 1 and 2, and we're going to look at the older population and the younger population. So first, let's look at the younger population, 18 to 55 years of age. That's what we're seeing here on this graph. And the two graphs up top are vaccine number 1, and the one here on the bottom is vaccine number 2. And just at first blush, you can see here that vaccine number 2 seems to have less in terms of side effects. Now, the other thing that I should mention is that they are looking at making this a 2-step vaccine process, with the first vaccine being given and then the second one being given on day 21, or 3 weeks later. Interestingly, also, this vaccine would have to be refrigerated in very tightly controlled refrigerators. And so here the first column is the first dose and the second column is the second dose. So let's take a look here at vaccine number 2. You can see here that at least 60 to 70 to even 80 or 90% of these patients had some sort of pain at the injection site. That's not too unreasonable. But in terms of redness, swelling, either on the first dose or the second dose of the second vaccine, that it doesn't look like there was a lot in terms of side effects. It seems as though the 30 microgram dose gave the most in terms of side effects. If we look in the older population, 65 to 85, once again, in the vaccine number 2, there seemed to be less side effects than in vaccine number 1. And once again, the biggest problem was pain at the injection site, but in terms of redness and swelling, we didn't see much there. What about more systemic effects? These are the kind of side effects that we're actually looking for a little bit more carefully. Once again, let's look at the second vaccine that was being developed. That's B2 here on the bottom 2. Fever, fatigue, chills, both in the first dose and in the second dose, were much less than that in the first vaccine that was being developed. And that dose 2 seemed to give higher side effects or higher incidence of side effects. The biggest one in terms of incidence was fatigue in both the first dose and in the second dose, ranging from about 40 to 70%. If you look at participants in the 65 to 85 years of age group, you can see how the second vaccine, or B2, had far fewer side effects than B1. So you can see the conclusions that the authors came to. They said the safety and the immunogenicity data from this United States phase 1 trial added to earlier interim safety and immunogenicity data support the selection of B2 for advancement to a pivotal phase 2-3 safety and efficacy evaluation. And that's exactly what they did. A press release about the preliminary results came out about 5 days ago. And they had enrolled 43,538 participants in this randomized blinded trial. And so what they decided is they did some calculations, and they figured that when they had heard that 94 confirmed cases had occurred, they were going to take a peek at the data to see how it looked. What they were looking for was to see how many of these patients who got COVID had gotten the placebo versus had gotten the actual vaccine. And what they were able to conclude based on the data from those first 94 confirmed cases of COVID-19 was that the vaccine B2 was found to be more than 90% effective in preventing COVID-19 in participants without evidence of prior SARS-CoV-2 infection in this analysis. And what they're actually asking for now is to continue the study to go up to 164 confirmed cases to see if this holds up and if there are other data points that can be made. That's important because if they were not to do that, if they were to end the study, it would be considered unethical to randomize someone to a placebo, given the fact that the vaccine is so effective at preventing COVID-19, a potentially deadly disease. And here is the press release announcing these preliminary results. We don't have this in a peer-reviewed publication as yet, but I'm sure it's coming. In terms of what's actually going on biochemically inside these people who get this vaccine is very interesting. And there are some hints because if we look at some earlier trials with this very same vaccine in monkeys and in mice, we see some very interesting things. And this was a paper that was published in Medical Archive, which we will put in the description below in terms of the links. What they did is in this study, before they did the phase 1 trials that we just reported on, they looked at how this vaccine candidate behaved in monkeys and in mice. And so they say here that this LNP formulated nucleoside modified mRNA that encodes the spike glycoprotein captured in its pre-fusion conformation, which is what you want. You want it to look exactly as it does on the SARS-CoV-2 virus. So what they found was that these neutralizing geometric titers were 10 to 18 times that of the SARS-CoV-2 convalescent human serum panel. And not only that, but this very same vaccine that they gave in humans later on, when they gave it to macaques and to mice, they found that their Th1 type CD4 positive and interferon gamma positive response was elevated, and a CD8 positive T cell response was also activated. And all of this points toward a wide and diverse immune response. And one has to figure whether or not this is the reason why it seems, at least on preliminary data, that this vaccine is 90% effective. So let's talk a little bit about the vaccine. Here you can see these lipid nanoparticles that contain the messenger RNA with the code in it. So if we look at what we have here with the vaccine from Pfizer BioNTech, they say it's effective at preventing COVID-19 at around 90%. Now realize the way these endpoints worked was that 7 days after the second vaccine dose, so at 28 days after the first vaccine dose, study subjects were asked to contact the site coordinator if the patients had any symptoms of COVID-19. And if after evaluation it was likely that they had COVID-19, these subjects were given a COVID-19 test. The study was interrupted when the number of subjects with COVID-19 positive test results reached 94. When this happened, they unblinded the 94 so they could see who did and who did not get the vaccine. It was at that time that they released some preliminary results showing that the vaccine was about 90% effective. So some interesting points about that is that really there are no asymptomatic subjects in this result cohort. We don't know what this does to asymptomatic patients. And furthermore, we don't know how long this effect lasts either. So those will be questions that have to be answered down the line. Of note, the Data Monitoring Committee reported that there was no serious safety concerns. The other question is, does it stop infection? That's important because this vaccine may be just important in the individual that gets it at preventing them from getting the disease COVID-19. But the question is, does it actually stop infection with SARS-CoV-2? That's important because if it doesn't stop infection, then you can still infect people with the virus and it can still multiply exponentially. But if it has a sterilizing effect, then it's potentially possible that you could block the spread of the disease. That is still up in the air and a question at this point. Now, a couple of numbers that you should know. Pfizer believes that they should have about 50 million doses by the end of the year. And they are working right now to try to get emergency use authorization. They also say that they should have about 1.3 billion doses by next year. You need to realize that there are 2 doses. Both doses need to be refrigerated in special refrigerators that keep them at negative 70 degrees centigrade. And the question is how much is this all going to cost? Well, they're saying that per injection, the cost will be about $19.50. But there has been some reports that they are working with the U.S. government, and for those in the United States, it should work out to be free. Now, also remember this, too, is that apparently Pfizer did not take any government money in the production of this vaccine, and furthermore, Pfizer is looking at producing this vaccine worldwide. So this vaccine does have some promise. I would be very interested to hear what you all have to say about this vaccine. It's one of 4 of the vaccines that we're going to do a deep dive and look into. I think this one is interesting because of its novel approach using messenger RNA. Of course, there's another vaccine candidate that is doing that as well, and that's, of course, the Moderna candidate, which we'll talk about coming up next. Keep in mind that no mRNA-based vaccine has been approved by the FDA. But this particular candidate is interesting because we have the preliminary safety data in, and it looks as though we have the preliminary results of a report about the efficacy data. Of course, more of that is to come when it gets published. And so people can start to make a decision about if this vaccine becomes available and gets emergency use authorization, and when it becomes available, if they would decide to get this vaccination. And don't forget to visit us at medcram.com for continuing medical education. Thanks for joining us.